Namaste. Vishwa Ayurveda Parishad Dravyaguna Sambhasha. Today is the 37th webinar. We are conducting the 37th webinar today. We are celebrated Vyasa Purnima also in the same month, in the last July 2010th. And we are conducting the Dravyaguna Sambhasha with CMA with Professor Vidyaguru Rammanohar sir, and all the distinguished guests are here. All Dravyaguna faculty members are also here to hear the Rammanohar sir's lecture. Just I will introduce Rammanohar sir in very short, because he is just like Vasudeva Kutumbakam for everybody. He is just like a referendix for every Ayurveda scholars. Rammanohar sir is born on 3rd September 1967. He finished his BMS from Coimbatore Ayurveda College. Because it's almost all the Ayurveda gurus are from Coimbatore Ayurveda College, and Rammanohar sir also learned Ayurveda and finished Ayurveda in 1991 from Ayurveda College, Coimbatore, and he finished his postgraduate degree from our college, that is Ellen Rao Ayurveda College. And after that, because he's born with the research, several research papers, as well as several research things he has done. He started his research from 1991 in the LSPSS as a research coordinator. From 93 to 97, he worked in FRLHD Bangalore. And from 97 to 200, he worked in Ellen Rao Ayurveda College, Koppa, as a R-Mark newsletter, as well as he done his MD degree also. He has multiple with lecturer also therein. And from 2001 to 2002, Reliance Health Insurance, he is part of that as a director. And from 2002 to 2003, he worked in AVN Madurai as a main research head. I was also part of that during his participation there. And from 2003 to 2012 in AVP Trust, because he is well known. Krishna Kumarji sir is always treating Ram Manohar sir as an Ayurveda guru, and always he is whenever any research project is there, he will give it to the Ram Manohar sir. He worked as a director in the research head, as well as in from 2012 to 2016 in AVP research. He worked as a head. From 2016, he is working in that as a director in Amrita Center of Research, and many things he has delivered. Because I am very proud to say, in almost all national as well as international any seminar is there, Ayurveda is concerned, he's one of the guest speaker there. And he traveled almost all the countries for this Ayurveda, because in his Kanakana Gulu Sharade, and one Kannada song is there. In the same way, I can able to say in Kanakana of Rammanohar sir's blood, Ayurveda is there. That's why I'm not taking much time for introducing him, because he is Ayurveda. He bonds Ayurveda in just like yada yada hi dharmasya glanir bhavati bharatah abhyutthanam adharmasya tadatmanam srajamyaham. For the revive of the Ayurveda, Rammanohar sir is present in presently in the India, just like because you can hear with many YouTube lectures on Ayurveda and revival of Ayurveda and Ayurveda research with his YouTube lectures. And I am welcoming Dr. Rammanohar sir for this presentation today, sir. Namaste, sir.
Namaste. Good evening. Thank you, Dr. Vinayak Bhatt, for that warm introduction. I still consider myself as a student of Ayurveda. It's a lifelong journey, and whatever I have learned in these many years, I'm happy to share and discuss so that we can develop this knowledge together. So my pranams to all here, Professor Anil Shukla sir, Dr. Vithi Bapna, some of the names I can see here, and everybody else. Thanks to Dr. Vinayak, he's like my close friend. We lived together for many years in Koppa together. And that is instance I could not refuse to be here on this platform today. So I hope I will be able to do justice to this topic. I'll share, on the basis of my own experiences in doing research in the field of Ayurveda. I can say it has been always a challenge for me to see, how we can bring in modern methods of research in Ayurveda, but at the same time preserve, the unique holistic approach of Ayurveda at the same time. So this is the biggest challenge. If you want to go purely in the traditional way, that path we can follow. Or if we want to go in the modern way, we can also go in that direction. But if we want to combine the two without one affecting the other, then it is challenging. I have a small request. If at least a few of you can switch on videos, it will help me to bring more focus in my discussion. Because as you know, this is one of the most challenging things, to speak to a blank computer. After a little while, your focus goes, and sometimes the ideas don't come in a creative way. So it's always good to see at least a few faces. Thank you so much. I know that everybody may not be in a position to switch on their videos. I hope my screen is visible.
Yes, sir.
Yes, sir. Yes, sir, completely.
Yes, sir.
So the topic for today's discussion, what was actually given to me, so I did not modify it: methodologies for research in Dravyaguna, challenges and innovations. I know we don't have too much time, so it's a very vast topic. What I would like to do is give some pointers for further thought. So rather, I would like to provoke thinking rather than give, definitive suggestions. So I'll be happy if people are a little also uncomfortable, want to ask more questions or think more. That is the purpose of any discussion anyway. We have to think. The thought process has to be kindled. Especially in short presentation, that is the only thing that is possible. Anyway, I don't think I should give too much of background and waste the time. So now we'll go into the topic. So when you think about Dravyaguna, even when I discussed about this talk, some people asked me, is there really Dravyaguna in Ayurveda? Where is this coming from? It's not a classical term. I think that debate is not relevant anymore. We have Dravyaguna in the academics. I also finished Dravyaguna for my PG, and we have to deal with it. And we cannot say that there is no classical perspective on Dravyaguna, because it is mentioned, Dravyam hi gunakarmajnam samyak sampradipadyate. Because without the gunakarma jnana of a dravya, we cannot use it properly. So two things are there in Ayurveda that I think will define the framework for Dravyaguna. One is the gunakarma jnana of dravya is very essential. That has been clearly mentioned. Not only gunakarma, nama rupa guna jnana is extremely essential, but we must also give emphasis to the point Charaka has made, Tishtatyupari yuktijno dravya jnanavatam sata. That yukti is actually more important for bringing Dravyaguna jnana to fruition. So I want to say that this is one thing we have to bring into the field of Dravyaguna, that it, rather than making it a non-clinical drug-oriented subject, we have to bring it into the clinical context. Then Dravyaguna has classical relevance and meaning. When yukti is also involved along with it, when it is looked at it from a clinical perspective. So mere knowledge of properties is in this framework I'm going to discuss how we can bridge traditional methodologies of understanding Dravyaguna karma with modern, cutting edge researches. in such a way that we preserve the epistemology of Ayurveda and strengthen it rather than. So that's the framework I'm going to discuss today. So it's a well accepted Ayurvedic principle that Dravya is understood through its gunas and karmas, and this is a concept that is deeply embedded in classical thought. So we can say that Dravyaguna is not only the knowledge of guna and karma, we cannot say it is just pharmacology or pharmacognosy, but also its clinical applications. That is why Dravya is one of the Chatushpadas. So in our classical text, we have looked at Dravyaguna from a clinical perspective, not just as a today's idea of pharmacology. We shouldn't translate it as pharmacology. It has that clinical dimension. That is the additional thing that we get from when we look at the classical literature. So Dravyaguna is actually a bridge between pharmacology and clinical medicine. That is a very important point that we have to emphasize. We should not separate it from clinical medicine. It is a bridge. So that is why in the Chatushpada it is there. That is why Charaga said, Tishtatyuvari Uktijno. That does not mean Dravyaguna is not relevant. That never the Acharya has mentioned. But it had to be brought into a clinical context. So framework-based research can ensure that Ayurveda principles can guide study design. So we have to use all modern methods keeping this framework in mind. So now if you look at it, that's what I'm going to say. I'm going to compare the classical approach and the modern scientific research and show where the challenges and gaps are and how we can integrate them to develop Dravyaguna into a clinical oriented discipline. So just like, Shariram, we have divided it into Sharira Rachana and Sharira Kriya, which is actually against the Ayurvedic principle of understanding the body as a functional whole. Our idea was Shariram, and our classification was Garbha Shariram, Srishti Shariram, Prakriti Shariram, Dosha Shariram, Sankhya Shariram, Vikriti Shariram, Ityatra Janma Maranam Yada Samyak Udahridam, Sharirasya Tadasthanam, Sharira Vidam Uchchita. We had a comprehensive understanding. Similarly, in Dravyaguna, from Namatjnana, Rupajnana, Gunajnana to Yuktijnana, we had a very comprehensive. So that should be the foundation for all modern research in Ayurveda. We shouldn't back translate modern terms and create a Sanskrit. So because there is pharmacology in modern medicine, we want to create an equivalent in Ayurveda as Dravyaguna. That is a mistake. What we have to see is we have to begin with Dravyaguna and see which all branches in modern pharmacology can be integrated with Dravyaguna. Then the result is different because we will get not just one correlation. We will have pharmacology, clinical therapy. Everything will come within the purview of. So Dravyaguna can actually create a synthetic approach of blending many disciplines because everything in Ayurveda is multidisciplinary. There is no compartmentalized discipline. If you take Dravyaguna, it gives a multidisciplinary perspective. If you take Shariram, it gives a multidisciplinary perspective. So many things in modern medicine, many subjects will actually come within that purview. So we should look at Dravyaguna also like that. So now if you look at the classical approach, we have so many theoretical frameworks. The Panchavidha Pariksha of Rasaguna, Virya, Vipaga, Prabhava, Karma, Vishesha and Anubandha Guna, Nigandu-based comparative studies, and empirical knowledge from classical texts and clinical observations. Our Dravyaguna has been enriched by all these aspects. So the strengths are that it is holistic, it is experiential and very contextual. It was very dynamic. Region-based Dravyaguna, Khandus were there, person-based approach. But the problem is that today, in today's world, when we present Dravyaguna, some of the issues that people say is the lack of standardization. Most of the descriptors are subjective. Quantification is minimal. Like if you ask about the gunas, Ushna Guna, 10 Dravyas are having Ushna Guna. How do we distinguish between the Ushna Guna of one Dravya from the other? So when you look at it in that way, this theoretical framework has some shortcomings the way it is presented. Now, if you look at modern drug discovery approaches, now we contrast it. Modern methods are target-based drug discovery. You're looking at molecular targets like enzymes, receptors, high throughput screening of everything is based on breaking down the substances into chemical constituents. And identifying the active principles. So there are so many such methods available now to pinpoint exact chemical compounds that are responsible for drug action, like crystallographic or computational model, molecular docking and in silico screening. We have phenotypic screening, which can look at disease models without even knowing the target. There is combinatorial chemistry. So it is quite complex. Now AI and machine learning has also come into drug discovery process. Then there are biologics and gene-based therapies. Then, of course, clinical trials and regulatory pipelines. In fact, in modern medicine, all research is drug research. So in essence, they study in clinical trials how a particular drug acts on a disease target. So these are the two contrasting frameworks that we have. The previous slide, I showed the Ayurvedic framework, and here is the modern. Now let us see the difference between modern and Ayurvedic, and then I will discuss as to how we can integrate the two. So this is the main thing I wanted to discuss in the short time that we have today. So if you look at the philosophical basis, modern drug discovery is reductionistic and it is target specific. Means if you take a plant, they want to identify one or two molecules and discard the rest of it. Whereas in Ayurveda, we have the holistic system-based Tridosha theory. So this is the question that I am asking. How can these two systems meet? If we use modern methods, will we not destroy our framework? Now, if I take Sarpagandha and just use Reserpin from that, will that be Ayurvedic? Is that the kind of research that we need? So source materials are isolated compounds which are synthetic or natural, whereas in Ayurveda, we have whole herbs and polyherbal formulations. Discovery purpose is lab-based, high-throughput screening, AI, animal models, whereas in Ayurveda, it is empirical, centuries of observation, textual documentation, maybe a lot of intuition, tapas, observation of animals. The whole methodology is different. Knowledge origin is very recent in modern drug discovery. In Ayurveda, we have thousands of years of documented knowledge. Evaluation in modern drug discovery is target-specific outcomes. It's biomarker-based, whereas in Ayurveda is multi-dimensional rasaguna, virya, vipaga, prabhava, which are systemic properties. They are not molecular or component-based, and they interact with the body also in a systemic way, on tridoshas, on dhatu system, not on single molecular targets. So validation method of modern is randomized control trials, whereas in Ayurveda is clinical experience, Anumana, Yukti, Pratyaksha. Formulation focuses a single active molecule in modern drug discovery, whereas in Ayurveda it is synergistic multi-drug herbal matrices, what we call as a yoga. Everything is a yoga or a combination. Ayurveda is a science of molecular combinatorics, and Ayurvedic medicines are molecular cocktails, if you were to explain it in modern scientific language. Now, in modern drug discovery, it is standard dose for the whole population, whereas in Ayurveda, it is personalized, prakriti-based treatment. So, end goal is disease eradication or symptom control in modern system, whereas in Ayurveda, it is restoration of balance, long-term well-being. So, I hope you can see the contrast and differences. So, now my question is, can these two systems at all meet? If you look at this chart, they are very diverse. And what are we doing in our PG studies, PhD studies? Have we really thought about the real bridges for integration, or are just for the namesake of making Ayurveda scientific? Are we really engaging in meaningless research? This is my question. I would have loved to pause and get some responses at this stage, but I know it will be very cacophonic on this online platform. So I'm not. So if it was a live thing, this is a point I would have taken some time for, feedback and discussion. But
Sir,
we are not.
we will have discussion after your lecture. So all the Dravyaguna stalwarts available. So we will have discussion after your lecture.
Okay, fine. So now the challenges with the current methodologies are epistemological mismatch. That's what I want to show. Epistemologically, Ayurveda and modern science are different. So, and in Ayurveda, standardization may be difficult because we are using the herbs as a whole. We cannot say that each ingredient should be present to a precise quantity. It will be varying according to region, season. It may not be exact like 500 mg paracetamol. We can say 50 ml of kashayam, but within that 50 ml, how many chemicals will be there? What is their relative percentage ratio? To standardize this may be very, very challenging and difficult. Now, when you look at gunas, Ayurveda explains all the dravyas in terms of their gunas and karmas. How can we compare this with modern chemical profiles? A one-to-one correlation is not possible. So when we say in Ayurveda, in modern medicine, there's an anti-inflammatory effect, is it just due to Shita Virya, or is it due to the combination of many other substances? Is there a consistent pattern that we can see between gunas and chemical profile? These are all questions that are not very easy to answer. Now the thing is, there is inadequate documentation of clinical pharmacodynamics from Ayurvedic perspective. And then the biggest challenge that I've seen is, if you want to use modern equipments to understand Ayurvedic Dravyaguna clearly, unfortunately, the simple methods are not so suitable. More suitable methods are very complex, like high throughput screening. If you want to get the complete profiling of a herb, we'll need to go for high throughput screening. We may need to go for high-end equipment, which makes it really very, very expensive. So generally what we try to do are to use the simple equipments, which are more good for fractionation and separation and not so suitable for Ayurveda. So now we know, we looked at the Ayurvedic framework, we looked at the modern framework, we looked at the differences, and we looked at the challenges. Now I am coming to what are we doing in actual practice today. We are doing a little bit, some of which is meaningful, some of which is not fully meaningful. I think Dravyaguna will take a new direction when we make the bridges very carefully, so that according to the Ayurvedic theory, according to the understanding, we will be able to use modern methods more meaningfully. That is the future. Now we are using modern methods for botanical identification, authentication, DNA barcoding, macroscopic, microscopic methods. These are helpful. Definitely for industry, it is helpful. We can now identify a plant more accurately because so many technologies are available. And I don't think this is any harmful at all, because specific species identification can be made more robust. Phytochemical profiling is done. We are doing HPTLC, GCMS, LCMS, NMR. This has some limited utility. With this profiling, we are not able to explain exactly how Ayurvedic drugs work, but we are able to use it for, again, for standardization, for quality control. We are able to identify some marker compounds. We are able to say maybe some of the active compounds are working in a particular way, but not the complete picture. Because now if you take Brahmi, it has got many components, bacosides, multiple components are there, and all of them in a combination actually work on Brahmi, in Brahmi. And Brahmi, many of these compounds are polar, some are non-polar, active compounds are lipophilic. And what we are finding is when you give Brahmi, it may not even cross your intestinal membranes and reach the bloodstream. If it reaches the bloodstream, first pass metabolism, it will be destroyed. Very little may even cross the blood-brain barrier. So when you use Brahmi as Gridam, and when you do Nasya, it may cross the blood-brain barrier. Otherwise, swallowing Brahmi tablets or Brahmi Swarasa may have no action at all. So to understand this whole complexity, simply doing an HPTLC or HPLC will not be enough. In my lab, we did a study. One of our students did it, gave Nasya of Brahmi Grida, and in 6 hours, we found bacosides had come into the bloodstream. So we know what is the time window. If we do these studies based on Ayurvedic principles, then only it will be helpful. Simply doing HPTLC, simply doing LCMS, and showing what are the compounds or components will not explain Ayurvedically why and how that drug is working. We need to be able to use modern science to explain why Brahmi is preferred as a Grida, why Brahmi Nasya is mentioned, what is the modality and mechanism of action. Can modern science actually explain this? Then we have in vitro, in vivo studies, cell lines, animal models. Here there is a big problem in a cell line study. See, we now in our lab, what we are doing is we are digesting the drugs using human digestion models and then administering in cell lines, because the drug will never reach the cell in the form that we are ingesting. Our digestive system will, so without using digestive simulators, if you study these drugs, it won't give an idea of what's happening inside the body. Similarly, when you use animal models, the problem is in Ayurveda, we have Prakriti-based, Agni-based, so many variables which are differing from patient to patient. Whereas when you use an animal model, we cannot look at these differences at all. It is one standardized dose given to all animals. We don't classify the animals into subgroups or subtypes of the disease. So what is studied in animals cannot be easily translated into humans. I'm not against animal studies. I think Ayurveda was one of the earliest systems to look at toxicity of drugs using animal models, because when they had doubt whether food is poisonous, they used to give to animals, which is very similar to toxicity study today. And zoo pharmacognosy was used for discovery of medicines. Today it's called zoo pharmacognosy. In olden days, they used to observe how animals are taking certain drugs when they get sick. Today, some months back, we read an article about an orangutan, picked up a leaf of a particular plant, chewed it, and applied on a big ulcer on its face, and it healed. This has been documented and published as a research paper. So, Atharvaveda also says the mongoose knows the medicine, the owl knows the medicine. I am learning from these animals. So all animal models by themselves are not problem, but the way it is designed today are for modern drug discovery. We have not adapted it for Ayurveda. So many of the animal-based studies today are not giving insights into Dravyaguna. Then there are pharmacological assays, anti-inflammatory, antioxidant, adaptogenic. The problem here with these models are, I know in PG, PhD, even our college, a lot of people are doing it. But they look at one isolated mechanism. They don't look at how the drug works on the whole system. So when you give it in a patient, it may not mimic. And that is why many of the Ayurvedic medicines are doing sometimes interesting findings in preclinical models, but they are not being replicated in humans unless we give it in a clinical way. Then the clinical verification approach, which is actually more needed in Dravyaguna, unfortunately, because it is not considered strictly as a clinical subject, this is a little bit, lacking. That perspective. So Dravyaguna should get integrated with Kaya Chikitsa-like approaches. We have to clearly define how the two systems will differ. But I think in Dravyaguna, we need to bring more clinical context to be able to understand Ayurvedic principles clearly. So now, with this background, I want to give now some suggestions on how we can bring in integrative methodologies. So if you co-apply Ayurvedic parameters and modern tools together, then we can develop new methodologies which we can call as integrated Dravyaguna research methodology. One example is you can correlate Guru Lakhu with digestibility index, caloric load. Actually, I don't have time. I'm working with some of my colleagues to develop what we call as a Guru Lakhu Guna index, a quantifiable index, where we look at multiple parameters, measurable parameters using modern science, give it a weightage. So it is, you measure all these quantifications, about 10-15 such parameters are there. Give it weightage, and then every substance that we use get a Guru Lakhu score. So with that, you can quantify the Guru Lakhu Guna. Actually, Guru Lakhu is to be understood in two ways. One is ease of digestibility. Another is whether it will add to your weight or reduce your bulk. Yasya Brahmane Shakti Sa Guru, Yasya Langhane Shakti Sa Lakhu. And Guru Lakhu in terms of how Agni will deal with it. Guru Nam Ardha Sauhityam, Lakhu Nam Nadi Trupta. There we are looking at how Agni will. So both have to be combined. And we found that very interesting findings. So we validate this score with known substances so that till the tool develops a sensitivity to match with what is described in the texts. Once that is achieved, which means the score given by the tool model matches with the description in the text. Then we know this tool has become sensitive. Now the same tool we can use to unknown substances, which are used in modern life. We can give them Guru, Laghu, scale, scores. So this is a very interesting possibility that if you use the modern methodologies of assessments within the Ayurvedic theoretical framework, they become very powerful. We can objectify many of the Ayurvedic parameters. Another approach, integrative approach, is something which Ashok Vaidya had talked about. It's called the reverse pharmacology approach. And unfortunately, in the Ayurvedic field, we have not taken it up very seriously or defined it from an Ayurvedic perspective, where we start from clinical efficacy. Now, in our PG, PhD, we should make it a point that in Ayurveda, somebody should start a study only based on clinical observations. Because clinical validation is more important for us. Something works clinically, and we are going to work back to find and validate its Dravyaguna, especially for known things. When something is already described in the text, which we are using in clinical practice, our starting point should be clinics. If it is a new drug or a new substance not mentioned in our literature, then we should start from the drug. So this should be made as a regulation. Because what we are using for centuries, what we are using today, what the students are observing in the clinic, why do you want to start from a preclinical framework for that? So that is why people like Ashok Vaidya coined this term, which is very specific in the context of Ayurveda, called reverse pharmacology. Then we have something called network pharmacology and systems biology, where we can look at multi-compound, multi-target herbs. Chinese medicine has already started looking at this, and they are able to understand how multiple components in a drug can upregulate, downregulate groups of genes, how it can produce systemic effects in the system. And this is where I said some funding, support, instrumentation is necessary. Otherwise, network pharmacology will not be possible. And now, with artificial intelligence and machine learning, we can actually mine Nighandus for property clustering and prediction. A lot of tools can be developed. Prediction models can be created, which we can validate through experiments till the AI learns and becomes capable of more and more accurate prediction. But I want to give a warning that we should never replace our mind or our intelligence with AI or ML models. It should be only a clinical decision support system. That we should, unfortunately, today even ChatGPT we are using to do our work. That shouldn't be there. It can be used as a tool. Now, high throughput screening approaches where we can bring omics, genomics, proteomics, metabolomics, because if you look at concepts like Tridosha, Tridhatu, Saptadhatu, Siddhanta, they are actually multi-omics approach in Ayurveda. We're looking at all these levels of genomics, proteomics, and metabolomics. Ayurveda has explained drug action not on one single molecular mechanism, but across this whole multi-omics framework. We can use what is today called as QSPR and QSAR models, like quantitative structure property relationship and quantitative structure activity relationship, because these are computational modeling techniques which are used in drug discovery and chemical research. Using this, the property of biological molecules and activities can be predicted. So in Ayurveda, this can be used for modeling complex interactions between various substances in Ayurveda. So large-scale libraries of Ayurvedic phytochemicals can be screened against disease targets. I tried something. There is this plant Desmodium gangeticum, which is in South India we are using. In North India, another plant is used as Thira. And in Vagbada, it is mentioned Hirsthe Paya Sthira Moolam. When your heart is affected by Vata, Sthira Kashaya, Shira Kashaya has to be given. So when we did the phytochemical profiling to find out using some AI tools, what are the drugs and substances that are likely to work on the heart and which extraction is better, water extraction or fat extraction or milk extraction. The AI was able to predict that milk extraction is most optimal for that sthira to actually work on the heart. So this is the kind of So that computational model was confirming what the Ayurvedic texts had said. So that chemical, so in this way, using these modern techniques, we can better understand, because in Ayurveda, we had that understanding that sthira had to be cooked in milk in order to have that optimal extraction of the polar, non-polar compounds to target the heart. So those things can be validated. In this context, I have to say about network pharmacology, which is a systems level approach to understand how multiple compounds act on multiple targets and pathways simultaneously. This is the complexity of Ayurveda. So here we are integrating bioinformatics, cheminformatics, pharmacology. I don't need to talk much about this. You can read more. So relevance to Ayurveda, Ayurvedic herbs and formulations are multi-component, multi-target. So because they are yogas, ganas, varga, yoga, gana. Varga means classification. Yoga means combination. Gana means computation. So the word gana in Ayurveda means many of these formulations were actually computed, not just combined. Yoga is a simple combination, whereas gana is coming from ganaka. What computer we call as a ganaka yantra. So it is computational. So we can map phytochemicals to biological targets and pathways. We can validate traditional indications of herbs using pathway enrichment analysis. Synergistic effects can be, studied. Active principles and their interactions with disease networks. So understanding the complexity of Ayurvedic medications and combinations can be better understood through network pharmacology. So for that we'll have to build Ayurveda specific herb, compound, target, disease databases. We have to bridge, concepts of Ayurvedic pharmacodynamics with molecular mechanisms, and in this way we may be able to create, new. What I have as a vision in my mind is that Dravyaguna will help us to evolve, an integrated pathway where Ayurveda medicines will be understood as molecular cocktails based on complex computations of molecular combinatorics. This seems to be a big possibility when we bridge Ayurvedic and modern methods together. Then we have ethnopharmacology and participatory research. This is very, very important because local knowledge, what people do, the Loka and the Veda, which our tradition talked about, tribal uses, contemporary research, community-based herbal surveys. This is also part of enriching Dravyaguna because it enables to understand traditional knowledge in real-life contexts. Look at many region-specific Ayurveda uses, Loka Parampara, Gram Vaidya traditions, and then, this makes the whole Ayurvedic pharmacopoeia actually very dynamic and evolving. And that is why we had the Nigandu tradition also, which actually did this exercise, this ethnopharmacology and participatory research. If we really look at how a Nigandu evolved, this has been the basis for that. So what I am talking about is a multi-layered research protocol, textual and traditional evidence as the starting point. Then the next level, pharmacognostical and botanical studies, and then doing a complete Ayurvedic profiling. This, I think, is a database that we have to create. I had put many examples in the coming slides, but I think I'll have to stop in the next few minutes because we have a short time, and there should be some questions and discussion. Otherwise, there's no point in this session. So once you have the Ayurvedic profiling of Rasaguna, Veera Vipaka, we know many dravyas. There are missing information in our Samhitas. We have to fill that. Some drugs, some gunas are not mentioned. Some the Vipaka has not been mentioned. So we have to fill up all that. We have not explained the drug action of every dravya based on it's Rasaguna Veera Vipaka. Why Haritaki is Vataka Bahara? In what terms can we explain it? We need to put it in a sequence, in a flow work. I mean, what you can say as a logical flow. All this information, when it is fed into AI models, then AI will be able to understand the Ayurvedic logic and link it with phytochemical and analytical work, create molecular signatures, disease target pathways that can explain what Ayurveda explains as pradoshahara action, not just one single molecular mechanisms. And with this framework, if we go into clinical observations or trials, then we will be able to make, I think, Dravyaguna Shastra more update. Without losing the Shastric framework, we can bring in modern scientific research. So I am not going into the other areas. As I told, what I wanted to point out here is that 3 approaches are possible in Dravyaguna. One is purely going in the classical way. The remaining of my slides, I have tried to point out, I won't be able to discuss that. What do we mean by going in the classical way? Continue to just look at Rasaguna, Virya, Vipaka framework, Tridosha theory, not bring in any modern knowledge. And that's the way we can, that's also not totally impossible. It can still be continued. But on the other hand, the next approach would be to completely use modern methodologies. And that is something which is, we are under a lot of pressure to do now. And there Ayurveda will get reduced to molecules, modern drug discoveries, evidence-based methods. That is another approach. But looking at today's pressure and the demand that we have before us, I think personally an approach that bridges these 2 extreme views would be most suitable for our future. And here the biggest caution that we have to exercise is to make sure that we follow the Ayurvedic traditional framework, because that is what gives the holistic perspective, and then use modern tools in such a way that it will explain this complex framework of Ayurveda rather than reduce it to just chemicals. And so this is the challenge. It's not an easy task. This is the direction we have to take. And here I would like to also point out that the Ayurvedic method of Rasaguna, Virya, Vipaka is now being recognized as what in modern science as something called as a chemosensorial approach. Chemosensorial approach means sensing the properties or chemical properties of a dravya or a substance through our sensory systems with our panchendriyas. So this is now emerging as a chemosensorial approach. This is something which we can bring into the framework of Ayurveda, where Ayurveda and modern science can be bridged without losing the holistic framework of Ayurveda. So I'm not going into those other slides, but I will just conclude with some of the frameworks that we could bring into Dravyaguna research. Is one, Rasaguna, Virya, Vipaka, Prabhava centric evaluation, where all the missing gaps in Ayurvedic texts can be filled. We have more elaborate Rasapanchaka frameworks for explaining drugs, including logics of pharmacodynamics and pharmacokinetics of Ayurvedic drugs explained in this framework. Dosha Dhatu Mala based pharmacological profiling, specific understanding of which dravya works on which dhatu, on which organ based targeting. How in Anupana and Sahapana influences the action of dravyas. Karma based drug profiling, like Charaka has given the 50 Kashaya, Panchashat Kashaya Kalpanas. Prakriti specific pharmacodynamics. These are all research frameworks that will preserve the Ayurvedic approach in Dravyaguna. Roganubandha and Hetu-centric studies, sequential drug use in Chikitsa Krama. We, if we are giving a particular drug, we do Dipana Pachana first, then Dosha Shamana, Shodhana, then we do the Brahmana treatment, or Agni Sthapana is done one after the other. So we'll need to validate this in an experimental model. And then Deshakala Satmya based variations. So these are all research frameworks where we can integrate modern methods of research, but still stick to Ayurvedic frameworks of Dravyaguna. So I think with that I will conclude, because we have only 8 minutes left. So there were many other slides where I was trying to go into detail on all these methods, but we won't have time to discuss that. So let me conclude here. Thank you so much for your attention.
Thank you so much, sir, for your insightful presentation. And now I request all the seniors present over here to please present their views, and I invite Dr. Shraddha Madam to please take over the question answer session and further discussion.
Thank you, ma'am. So it was a wonderful intellectual piece as always. We have many experts. For the concluding remarks, we have Nisi ma'am who has joined. We'll take her comments at the end. Are there any questions from the participants? If there are any questions, can you please pose a question?
Namaste, madam.
Namaste.
Good evening,
It was
ma'am.
really very wonderful, comprehensive presentation. Namaste.
Thank you for taking the time. I'm very honored by your presence, ma'am.
Hello.
Hello. Participants, can you please post any query, if any, of the chat box? Until then, we can take the comments, expert comments.
Please make madam.
May I request
Please make Dr. Tanuja madam, please comment on this and give your ideas on this presentation. Please, madam, go ahead. Tanuja madam.
At the outset, I would like to congratulate Rammanohar for it was very comprehensive, and I think you touched all the aspects of Dravyaguna research. Am I audible?
Yes, madam.
yes, ma'am.
Okay, thank you. And as we say in Dravyaguna, as everyone in the second year, as we understand, is any Dravya, and Dravyaguna, what we understand is in the three pillars, three areas. One is a Namarupa Vigyan, second is a Gunakarma Vigyan, and Prayoga Vigyan. For any Dravya, for example, Ashwagandha, which is now widely discussed. So Namarup, Gunakarma and Prayoga, and for Namarup, there are various, what you mentioned about starting from the authentication, identification, standardization, what are the substitute, what are the adulterations, and everything, all quality parameters, quality, safety, efficacy, what we understand. For Gunakarma, as you mentioned, Ras, Gun, Virya, Vipak, and to understand in the contemporary perspective is very important. And the Prayoga Vigyan is very important, as I mentioned that, as we mentioned that Karma Bistu Anumiyante Nana Dravya Shraya Guna. So when we say that Dravya and Guna has a Samavaya Sambandha, Samavaya is to Nishcheshtaha Guna. So without Guna, Dravya cannot exist, and Guna has no meaning. So in Dravyaguna, Karma may it be clinical, may it be pharmacological, preclinical, but the Karma Dravya research is without Karma Dravya, and Dravya Guna cannot be understood. So for us, Prayoga Vigyan in all forms, not only clinical, but preclinical, in vitro, in vivo, computational to pharmacological to clinical is very, very important, as we say, Dravyaguna. So in quality, one is a quality aspects, identification, Namarup, Gunakarma, but Prayog is also very important. As you mentioned, Vaidya sir, Ashok Vaidya, revered our Guru, reverse pharmacology is also one very important. To me, to all my colleagues who have joined here, Professor Anil Shukla, Shraddha Madam, Vidyapeetham, I can see Vinayak Bhatt, and Meenal Lad, Om Prakash Raut, and many, all are, we know, are very, what we can say, highly accomplished faculty. But to me, my teacher taught me, and what we have communicated, translated for last 35 years is Dravyaguna starts with the research. Pariksha starts with the Dravya Bhaishacha Pariksha from Charak Viman Adhyay 8. Idam evam prakruti evam gunam evam prabhavam asmin deshe jatyam anaya cha matraya. So I think Rammanohar ji, if we create a platform, computational AI-based platform, where we put all this Vaishya Pariksha, all important plants which are being used, highly traded, highly used with the clinical relevance. Maybe we put all this, I think we will get the template, which is a standard one, which is a Acharya, like a Shastra Sammat, and then we can try to understand into contemporary design. This was one addition I wanted to say. And the secondly, for Dravyaguna, if we create a consortium all across, like there are Shalakis, there are Shalya consortium. So to give something to best to our own fraternity, for our, because we say that one of my teacher used to say that if Kai Chikitsa is the brain of Ayurveda, Dravyaguna is the heart of Ayurveda. So if there is a like ischemia, then without Hetu Linga and Aushat, if Adravyabhut Chikitsa we cannot do. Only Daiva Papashra and Sattva Papashra will be there. Even for the money also, Ratna also we need to know Dravyaguna. But at least for the medicinal plants, what 8,700 Torah 222, 2022, which is codified, what I would like to urge to all of us, maybe on this platform, we select only 10, not more than that, only 10 Rammanohar, 10 plants which are high in demand, highly in clinical practice, and maybe we create a monogram, as we mentioned, internationally what we really require, Chinese medicine, or when there is a EU and in other countries also, FDA and others, we require a critically prepared, well prepared monogram, which includes everything, as you mentioned. So maybe 10 plants we start doing it, and we thoroughly understand, we thoroughly do the research, starting from the Ayurvedic understanding, starting from all these parameters, as you mentioned. And it is what Ras is not only chemosensory, what you say, but as we understand, Ras is more than that, as it is biophysics, biochemistry, molecular understanding. So even what we understand, we do it as a medicinal plants and clinical study. But to understand their mechanism in current perspective, the mechanistic study, we require pharmacological study, like animal models, only to understand the mechanism. And now there are many other models which we understand the mechanism, computational, bioinformatics, preclinical. So those are also well studied. Recently, I had been an opportunity to visit to one of the IITs where they use force field group of phytochemicals for the computational model and to 3D cell culture. So computational and the dynamics are completely changing, like it is 3G technology, now 5G technology, and the computer is becoming supercomputer, and we are using the iPhone is the 16 Pro model, and next year it would be something new. So the methodology and the tools, those are the dynamics. So those are changing, and we will get more and more refined version in coming years. But if we decide to screen at least 10 plants altogether, and we select what is the methodology, as you mentioned, starting from base is our classical idam evam prakruti evam gunam evam prabhavam, and then from namarupa gunakarma we plot our own thing, because if we are we know what we are very clear in our mind what is to be done, then we can adopt the conventional tools and technology which has the correlates, which correlates. When we say that the Raskul Virya Vipak, and as you mentioned, Rammanohar about do it with Rasguna Vipak and do it with the conventional tools. And if there is a similarity, that can be used for the unknown as well, Anukta Dravya as well. But that can be a separate research to study the Anukta Dravyas, to evolve how to assess Rasgun Virya Vipak. That is the fundamental research someone has to do. Someone has to really do. So these we can have the different what research questions, because when I say that I am working on Ashwagandha, but what is the research question? Whether it is a Namarup, whether it is a standardization, what is the best quality of Ashwagandha I use? So that complete different question. Then different on depending on the various research questions, if we collect together, that will become a complete monogram. Maybe 1 year as NMPV, we had launched And now NMPP is also launching as a campaign and just giving the funding for the different aspects of one plant. So maybe we do it from Dravyaguna at undergraduate, postgraduate, PhD, IMR, AMR, and we evolve. Like it is always, I say that 35 years when we started Dravyaguna research, and after that, we are the better version of our own self. So when I visited Jamnagar and I just reviewed my MD dissertation, I feel that how best enriched it was when it comes to the Tattva Shastra, because we were very much grounded and true to the science. But when it comes to the application, then I think we have crossed too many things. New things is coming, but that does not mean new things. Someone does not know that does not mean that we are lacking something. What is more important in Dravyaguna research is to understand our own tattva fundamentals, so the applications can be developed. With this, I congratulate once again Rammanohar ji. This is really excellent, comprehensive presentations you have created. Many research questions with the reductionist approach, with the whole system study. What is a conventional versus classical? Whether it is classical yet contemporary, tradition supported with technology. So many things. Maybe we can have 1 or 2 days workshop, or we can have a continuous course. You can create a module, Rammanohar ji, we can create a module for this capacity building. On each topic, we can have more deliberations. So with this, I congratulate and thank everyone, organizers, for having me here. I'm Meena Devgade, and I know that this group is doing the wonderful. Because of the time constraint, I could not join. But today, it was being a Sunday, it was a good opportunity that I could join. So, and I could listen carefully, because this topic is again close to my heart. So congratulations and thank you very much.
Thank
Very
you
nice.
so much, ma'am.
Thank you,
It
madam,
was
for all
honor
the
to have
suggestions.
to always have your expert comments. Thank you so much. And as you said, we need more of.
So we do invite madam for next month. Dr. Shraddha, do you
Yes.
invite Tamina madam for next month lecture?
Yes,
So
ma'am.
madam, you are invited for next month, last Sunday lecture. So please book the date and time, please. I will communicate up further on this. Okay.
Yes, we need your separate lecture, Tanuja.
Okay.
Yeah. And Rammanohar ji, it's a very good opportunity to hear you again. I'm Dr. Meenal Lad, sir.
Namaste, madam.
Namaskar, sir. Namaskar. Yeah.
Meena
Yeah, I
ma'am, we
think
can
this
have
is
your
a
comments.
very vast topic. More experts should come, and we should do brainstorming. Madam, what you suggested, workshop, monographs, are excellent ideas.
Yeah. Very
Because if we create some models, I fully agree. I mean, I didn't use the classical references because of lack of time, but all the frameworks were based on what Charaka is mentioned. I just tried to use an integrative
Yes.
language. So, thank you so much for this opportunity.
Thank you.
Thank you, sir. Thank you, ma'am.
I think sir has thrown the seeds to the PG guides to throw the more research. I think for the PG guides, he has given more seeds to now to grow. Thank you for that opportunity,
Yes.
for sir.
You changed our actually vision regarding the research. Like the Brahminasya, and you have to investigate the mycopene in bloodstream. That is the best parameter for the Brahmis, actually that chemical constant. Yeah.
Yeah, as
Very
Tanuja
nice.
mentioned, it is very multidimensional, and new technologies are coming. I think in the future, our complexity may be better understood when more complex tools are available. So what I have felt is that Ayurveda is actually very complex, but we are trying to simplify it using some of the simple tools. So hopefully we will all start using these new technologies in a proper way. And for that, more funding is needed, madam. I don't know what is your opinion on that.
Yes.
Most of the PGs department, they don't have the capacity to do these kind of work because they are very expensive. I think now the CCRA's initiative is very good, like the Spark and PG Star. At least some funding is being made available, but more funding may be required to be able to do these kind of research.
Yes, actually, what I feel with all Dravyaguna PG guide, maybe we have one brainstorming, and maybe there are two aspects for at the PG dissertation level to understand the research methodology and to conduct actual research as one piece. So maybe we do methodology as such, we can train all the scholars and actually our guides, but let us be more focused by next to low hanging fruit. What is our short term goal, mid-term goal and long term goal? So if we are united together, maybe we'll bring out something. Otherwise, what is happening? It is a repetition. It is a simple work. And what Kai Chikitsa is doing, again, we are doing repeating the same work. Maybe we have better opportunity, no one, anyone else can. And as not a single person, but we can do better than anyone else can. So if we join together, and then it is sort of a consortium, we can definitely bring out the impact what is this required in a short time. And we'll find out the funding part. Actually what I understood in these years is that we have to really work, we have to showcase the impact, and then the funding flow comes. Yes. Okay, we'll decide the areas and the research questions, I think.
Yes, ma'am.
It's really very good for us that Tanuja madam is come back again on home pitch.
This is our true identity.
Oh yeah.
Thank you. And it will take time, at least half an hour time in our next one. When the class can be this
So, sir, I
I
officially
never know every
extend the
second
vote of thanks now.
Yes, madam, please go
So
ahead.
I express heartfelt gratitude to Dr. Rammanohar sir for sparing his valuable time. Sir, it was a wonderful presentation. Thank you very much, sir, for being with us today in this VAP-Sambhasha. And definitely it will pay way for us, all the PG guides also and the PG scholars who has joined in a very good number. I take this opportunity to thank Professor Dr. Tanuja Nesri, Madam Director, ITRA Jamnagar. Thank you very much, Madam, for joining this Sambhasha and also giving very valuable inputs from your side. Thank you so much, Madam. I thank Professor Anil Shukla, sir, for putting all efforts to make this event successful. I thank Dr. Vinayak Bhatt, sir, Shraddha, Madam, Dr. O.P. Raut, sir, Dr. Meenal Lad, Madam, Dr. Rashmi, Madam, all for being here in this platform today. And I thank all the participants. Today there is a very good number of participants. I think so. I thank all the participants also for joining us today. And over to Dr. Vinayak Bhatt, sir, for the Kalyan Mantra.
We will pray because it is a Shravana Masa. That's why Sadyo Jatam Prapadyami Sadyo Jata Yave Namo Namaha Bhave Bhave Nati Bhave Bhavasvamam Bhavotbhavaya Namaha Vamadevaya Namo Jyeshtaya Namah Shrestaya Namo Rudraya Namah Kalaya Namah Kalavikarnaya Namo Balavikarnaya Namo Balaya Namo Balapramatanaya Namah Sarvabhutanamanaya Namo Manonmanaya Namaha Agorebhyo Tagorebhyo Goragoratarabhya Sarvebhya Sarvasharvebhyo Namaste Astu Rudrarupebhyah Tatpurushaya Vidmahe Mahadevaya Dhimahi Tanno Rudra Prachodayat Ishana Sarva Vidyanam Ishvara Sarva Bhutanam Brahmadipati Brahmanodipati Brahma Shivo Me Astu Sada Shivom Om Sahana Vavato Sahano Bhunakto सह वीर्यं करवावहै तेजस्विनावधीतमस्तु मा विद्विषावहै। ओम शांति शांति शांतिः। ओम सर्वे भवन्तु सुखिनः, सर्वे सन्तु निरामयाः, सर्वे भद्राणि पश्यन्तु, मा कश्चित् दुःखभाग् भवेत्। ओम शांति शांति शांतिः। Thank you to all, sir.
Thank you very much, everyone. Good night.
Thank you very much for nice
Just
mantra and Kaya mantra. Sir, please stop the recording, sir. Stop the recording. Like what, sir? So, thanks to Rammanohar sir. And start join today. I know it's a Sunday, being Sunday, everybody is in holiday mood. So, once again, and we look forward for your active participation in future webinars. Thank you, one and all.
thank you, ma'am. Thank you. It was nice word.